There are two types of flows when it comes to energy. And for the most part, there's cellular respiration and fermentation. Fermentation is a relatively inefficient catabolic process that leads to the partial degradation of sugars in the absence of oxygen. Glycolysis plus reactions that regenerate NAD plus by transferring electrons from NADH to pyruvate or derivatives of pyruvate. The NAD plus can then be reused to oxidize sugar by glycolysis, which nets two molecules of ATP by substrate level phosphorylation. There are many types of fermentation, differing in the end products formed from that pyruvate. Two common types are alcohol fermentation and lactic acid fermentation. In fermentation, the energy is passed through pathways in glycolysis and fermentation to lower energy molecules until the electrons are finally passed on to the acids and the alcohols. The energy that is lost in this process in passing these lower energy molecules is then captured by these electron carriers and used to make ATP. Fermentation is the process used by cells that cannot respire. For cells that can respire, they use cellular respiration. Cellular respiration is a more efficient and widespread catabolic process in which oxygen is consumed as a reactant to complete the breakdown of a variety of organic molecules. Similar in broad principle to the combustion of gasoline in an automobile engine after oxygen is mixed with hydrocarbon fuel. Food is the fuel for respiration. The exhaust is carbon monoxide and water. This includes both aerobic and anaerobic processes. Depending on if oxygen is the carrier or if oxygen is not, then it is obviously an anaerobic process. However, it originated as a synonym for aerobic respiration because of the relationship of that process to organismal respiration, in which an animal breathes in oxygen. In respiration, this energy is passed via the pathways found in glycolysis, the TCA cycle, ETC, to lower these energy molecules until finally electrons are passed to oxygen through aerobic respiration or other molecules such as something like a solute during anaerobic respiration. The energy that is lost in this passing to lower energy molecules is captured again by electron carriers, and this is used to make ATP. This is a much more efficient process than fermentation and therefore produces about 30 to 40 ATPs per glucose molecule in these pathways as compared to fermentation, where only about two molecules of ATP are created during this pathway.